Hello, welcome to the LabRoots webcast on our team science project that we entitle Neuronal Circuit Mechanisms of Memory Replay. This NIH Brain Initiative Team Science project is led by four of us, Yuri Buzhaki from NYU, Attila Loshonci from Columbia University, Mark Schnitzer from Stanford, and myself, Ivan Scholtes, also from Stanford. And I should add that this project was initially, to a large extent, conceived in close collaboration with the late John Lissman, whose ideas still guide us today. The general goal of this project is written here on the slide, and I'm going to unwrap it for you. So the general goal is to make the first attempt to fully understand a cognitively important event called memory replay during hippocampal sharp-wave ripples in terms of the detailed properties of the brain cells and circuits involved. So let's discuss this a little bit in more detail. What are hippocampal sharp waves? Why do we think it's a cognitively important event? And what does it mean to fully understand such an event? So sharp wave ripples, which we abbreviate as SPWRs in this presentation, are local field potential oscillations at high frequencies, 100 to 200 hertz. You see it on the number 1 on the left. These events can be recorded with an electrode placed, a relatively large electrode that we refer to as local field potential electrodes, placed into the hippocampal pyramidal cell layer in the CA1. And occasionally when the animal is at rest, you see these high frequency oscillations, the shortwave ripples. The shortwave ripples or ripples are often co-occurring, the fast oscillations often co-occurring with a slower wave that we call shortwave. And this complex together is what we refer to as sharp-wave ripples. They are generated at a very high level. Just as a first approximation, they are generated by fast GABAergic inhibitory interneuron and pyramidal cell interactions, where the pyramidal cell gets excited by input from the CA3, the presynaptic CA3 region through Schaefer collaterals. The excited CA1 pyramidal cells then drive local interneurons, and the local inhibitory interneurons then inhibit back on the pyramidal cells of the CA1. And this cycle, if you want, resonates at high frequencies at 100 hertz or above. These are the ripples. Now, why are these ripples important for memory? We know from several groups, and Yuri's group's results are shown on the right. We know that they are important for memory because if we selectively suppress these events, the spatial memory performance is decreased. So that's why we know that these events are not just there in the brain, they actually serve an important function. And importantly also, these events are not just there in the rodents, such as mice and rats, but they are actually present in all mammals that people have looked at. Now, what does it mean to understand an event like this? We borrowed, or we are guided by a set of criteria that people who work on simpler nervous systems in the crab and other organism, lobsters, developed a set of criteria that they thought we must meet to understand a neuronal circuit and its function. And you see the list there. So first of all, you have to identify the cell types in the circuit. You have to describe their connectivity and the distribution of the connection strength. We have to determine the intrinsic conductances of neurons, each neuronal cell type. We have to measure the overall output of the system. And then we have to determine how this output is affected by inactivation of individual cell types. And we can create a computational model incorporating the properties of the cells involved and their connections. And the critical test of understanding is whether the model can replicate or reproduce the experimentally measured output of the network. In order to arrive at this understanding, we use a set of advanced experimental and computational modeling techniques, some of which are shown here, and you will hear about them in more detail. So for example, Attila Losonczi's lab is using simultaneous two-photon imaging and electrophysiology in behaving animals in a multi-sensory context. Yuri Buzsáki's lab uses, for example, various optosilicon probes for large-scale electrophysiological recordings and optogenetic manipulations in freely moving animals. Mark Schnitzer's lab uses ensemble calcium and voltage imaging in behaving mice and uses various techniques that allow in vivo optical imaging across the entire dorsal surface of the cortex of the mouse. And my lab uses multi-scale computational modeling techniques to model and really understand hippocampal networks. So what are the big questions that we are trying to get at in this project? One of them is, how are sharp-wave ripples initiated? What are the external and internal controllers? What are the external, for example, what are the controllers that are external to the hippocampus? For example, the entorhinal cortex or some other brain structure that may be connected to it. And what are the internal controllers? For example, the circuitry of the CA3 or the dentate gyrus. How do they play a role in initiation? Another big question is the sequencing that occurs during sharp-wave ripples, and you will hear about this in the second presentation. And how are sharp-wave ripples terminated? What are the cellular synaptic mechanisms of sequencing and termination? How are memories stored and replayed during sharp-wave ripples? How is the content of sharp-wave ripples routed through distinct output pathways? And finally, we use theory and modeling of hippocampal networks during spatial navigation and memory replay to really understand how all of this happens in terms of the cells and synapses involved. I should say that the experiments are guided by theory and modeling, and the theory and modeling in turn is informed by the experiments. So this is really an interactive project between experiments and theory modeling. Now, this brain team science project is really a team science project in the sense that the various groups located at 3 universities are really interacting to come up with joint projects. And there's a lot of extensive added value, if you want, of this team science project. It's really more than the sum of its parts. Not just in terms of the joint projects that we do, the collaborations that occur, but also there is extensive technology transfer between the labs. There's free exchange of ideas and early feedback on projects, which are really important. We can discuss these, and we do discuss the early results with each other throughout the labs, and it's really helpful to guide how the research is going. Our postdoctoral and graduate student trainees visit each other labs, and they form important links with other postdocs and graduate students and interact with the PIs. All of this, of course, presents a lot of new networking possibilities and new collaborations that can actually take place even outside of this brain team science project with other labs in the States and outside. We also think that the data sharing that occurs between the labs is an excellent, if you want, laboratory for testing new data formats that allow the extensive sharing of data and computational modeling results across the labs as well as with outside labs. So without further ado, let me now hand over the presentation to Yuri Buzsaki for NYU. Thank you.
Hippocampal sharp wave ripples have the reputation of being the most synchronous pattern in the mammalian brain. As you can see in this figure, the LFP pattern recorded from the CA1 region of the hippocampus very nicely classifies various behaviors into two major classes. We can call them theta, or exploratory activity, or still activity associated with the sharp wave ripples that corresponds to the larger category of consumatory behaviors. It is visible from the figure that these patterns are strikingly different. When a sharp wave occurs, that corresponds to the depolarization of the apical and dendrites of CA1 pyramidal cells. And because of the stronger excitation coming into this region, the tug of war between interneurons and pyramidal cells produces a very fast, about 150 hertz ripple oscillation. So you can see on the figure here that this is the sharp wave with the dendrites, and here in the CA1 pyramidal layer, we've got ripples. Now, ripples have a reputation not only because they are very strongly synchronized, but also because of the spike content. In this next figure, I'm showing you 13 pyramidal cells, which are also place cells, which means that when the animal is going from left to right on this track, the sequential activity of these neurons will are correlated with the animal's position. These are called place cells. Now, the remarkable thing about this activity is that at the beginning of the journey, the same exact cells are also firing. And you can see it from here, they are firing exactly in the same order, but much faster in a much compressed way than during the journey itself. Even more remarkably, at the end of the journey, when the animal is again rewarded here and on this end and is involved in consumatory behavior, then the replay is reversed in the reverse direction. Now, this phenomenon has been named in by various terms, such as memory consolidation, planning, imagination, internalized deliberation, and others. And it has been investigated in many, many labs in various species, including humans. Now, over the past several years, many new questions arose. And I'd like to summarize the three major areas where I think we have to focus a little bit more. The first one is the specificity of the sharp wave ripple. The specificity, by specificity, I mean is that there is a extraordinary, on the one end of the spectrum, we assume that there is a very strong relationship between what one can see in the waking behavior, such as shown here, and the replay time that occurs during sleep or consumatory behaviors and in a compressed manner. So here, the colors are exactly the same as here. It's a very precise replay. On the other end of the spectrum, we can say there is absolutely no relationship between what happens in the waking state and what happens in during the sharp wave ripples. The truth is probably somewhere in between, and in various circumstances and various conditions, we see something in between these two extremes. So this is a task for the future to figure out under what conditions experience may change this attractor state and choose which one is activated and for what reason. The second issue that I'd like to introduce is that the modifications and the replay happens not only in the hippocampus, because these replay patterns are very powerful and they are affecting very large parts of the rest of the brain, including the neocortex. And then the question is whether the neocortex also has a similar replay. So what is shown in the left side here is the replay in the hippocampus during sharp waves. We can call it primary reading. And then the downstream reader structures, such as the neocortex, can do the secondary reading. And of course, it's not very clear whether how faithful it is from this pattern to this pattern. And the same problem can be also posed to the neocortex, as I just explained a slide ago in the hippocampus. Now, the further complication, of course, is that the neocortex and the other readers are quite complicated, and they have their own lives and connectivity, so they can read out various patterns. In other words, the same exact sharp wave ripple can mean totally different things to downstream readers. And these are shown in a cartoon here, that depending on the transformation function of the structure, the interpretation of the hippocampal ripple could be quite different in one area or another. Here is an extreme example. There are two major ways how you can read out the hippocampal pattern. One is through convergence. For example, all the outputs from the hippocampus can converge upon the same exact cell, such as can happen in the nucleus accumbens, or it can happen in the lateral septum. Or the other way around, which is the other extreme, is that this relatively compact hippocampus can be read out, or the content of the hippocampus can read out by multiple readers in a fan-out version. So this reader and this reader can interpret something totally different than a downstream reader here. So let me show you an example of the fan-in or convergence result. This is an illustration of the very strong convergence from the CA1 and the CA3 region or pyramidal cells onto the lateral septum, and you can see that they are overlapping quite a lot, and there is a nice topographical relationship between various hippocampal, subicular, and entorhinal regions in the lateral septum. Because of this strong convergence, there is also a ripple generated in the lateral septum, and many neurons are firing at the time when the sharp wave ripple occurs in the hippocampus. And the question here is that what is the interpretation of this downstream structure about the hippocampal output? And the answer is rather complicated, but can be simplified in a way by saying that there is not much information available for the lateral septal cells about the sequential activity. It doesn't matter whether the sequence is A, B, C, D, or the other way around, as it's shown by these analysis here. What the lateral septal cells really worry about is the total amount of excitation they receive from the hippocampal output. To summarize here, the lateral septal cells don't see and don't read the details of the sequential activity of the hippocampus, but they integrate the overall activity, that is, they read out synchrony from the hippocampal output. Now let's see the other example, divergence. A good example of the divergence is when we start from the CA1 region. The activity can go to the subiculum, from subiculum to the retrosplenial cortex, and then it fans out to the very large area of the neocortex. And so this is already a very strong connection, and we want to know what is the relationship between the hippocampus and a disynaptic target here, such as retrosplenial cortex. We can put in the electrodes in both structures, and what we can see is that there is a nice relationship during hippocampal sharp waves between the CA1 region with its own ripples and in the superficial layers of the retrosplenial cortex. In this little cartoon down here, you can see that many neurons can respond during hippocampal ripples in the retrosplenial cortex. The question is whether these patterns are identical, independent of the pattern of the CA1 output, or whether the CA1 output sends out various patterns, and then there is a differential response in each time that there is a different output coming out from the hippocampus. So this is done quantitatively by clustering the population vector, that is how and which order neurons fire in a given time window. And if we have about 100 hippocampal neurons, we can make these clusters. And in this particular session, we ended up with 10 clusters. So each of these clusters are shown here, and the bottom line here is that within clusters, the activity patterns is very similar, and across clusters, the activity patterns are quite dissimilar. And of course, the interesting problem here is that whether the downstream readers can discriminate between these artificial clusters or clusters that we made artificially. First, let's look at what happens in the hippocampus when we are zooming into cluster 1, 2, 3, and 8. And you can see not only the magnitude, but the neurons that are involved. These are the activity patterns are proportional with the yellow color. You can see that in this case, there are differences between these. It's not only the hippocampus that classifies these patterns, that's what happens in the hippocampus, but also the retrosplenial cortex. So for example, during cluster 1, you can see that initially there is quite a bit of inhibition in the retrosplenial cortex. During cluster 2 is an excitatory pattern, mostly in the bottom neurons, and then it's different in every single case. So this is a population readout. These are about 200 neurons here that are simultaneously reading out and discriminating pattern 1, 2, 3, 4, and so on. Now, the issue is whether there are individual neurons that are capable of doing that. And the answer is yes. So I'm showing you 1, 2, 3, 4 individual retrosplenial neurons, and each of the 10 patterns here, and each of the 10 patterns are also shown here on different number of trials. And again, the summary picture here is that there are neurons in the retrosplenial cortex that are tuned to 1, for example, number 7 cluster, and they respond exclusively when number 7, that is one of the particular patterns, is recurring. This particular neuron likes to respond whenever number 7 and number 9 categories are present in the hippocampus, and there are several other examples as well. So to summarize this part, different downstream structures and neurons, individual neurons, can read out different constellations of neural population patterns of sharp rectangular types. Now, this was a small structure, just the retrosplenial cortex, and of course the neocortex is enormous. So we would like to introduce new methods that allow us to read out how the entire neocortex interprets hippocampal outputs. In order to do that, we combine electrophysiology and wide field imaging methods, and this way you can image the entire right hemisphere, or almost at least the dorsal part of the entire hemisphere here. Here's a little result showing that depending how the neocortical patterns go down and up states relate to the sharp wave patterns, and their interactions can produce various interactions, and they are shown by the readout of this very complicated traveling patterns of activity. Now, obviously, this is a very crude resolution pattern. So in order to improve that, we would like to know what aspects of the hippocampal outputs are useful and important. And the next finding I'm showing here is that depending on the duration of the sharp wave patterns, the magnitude of the neocortical response shown by these red arrows varies quite a bit. In order to zoom into the single cells, of course, it would be good to have very large scale, high resolution techniques. And this is where we collaborate with the Schnitzer's group, because we have to rely on much better methods than the ones we have at the moment. So if the wider sharp waves have a stronger influence on them, we wondered, are they better, or when we have wider sharp waves, does it make any difference for the rest of the brain and perhaps for behavior? And indeed, what we found that there is a difference between short and long sharp waves, and the distribution of the sharp wave durations show a several order of magnitude distribution, and can be described by a log normal pattern. So this is a logarithmic scale, and this is the duration. And what we find is that this right tail of the distribution is important, because whenever the animal is in the novel environment, there are more longer sharp waves. The same applies also when we compare the sharp wave durations in tasks where there is a memory requirement or there is no memory requirement. When memory is required, the animal has to memorize something, then during replays, the sharp waves are longer. Even in the same task, we can ask what happens when the animal is performing correctly a trial or makes an error. And the answer is that on correct trials, the probability that the sharp waves are longer is higher compared when the animal makes an error. So if this is the interesting observation that longer sharp waves are better, so to speak, then we wondered what happens if we artificially prolong the duration of the sharp wave ripples. To do that, we express channel lobes in CA1 pyramidal cells, And whenever sharp waves occurred in a closed loop manner, we added an extra 100 millisecond. You can see here, that's the prolongation of the ongoing sharp wave ripple. To our surprise and happiness, we found that indeed, when we artificially, optogenetically prolonged the sharp wave ripples and added further content, spiking content to the ripple, then working memory performance was improved. There is a short summary here, namely that neural sequences of long duration ripples cover very large parts of the routes because the ripple duration seems to replay a longer route that the animal is planning ahead. And this is critical for memory, and even at the times when we artificially use longer sharp waves, then that route extension can be achieved. Now, to summarize what I had said in this presentation, we have to address the issue of primary reading of sharp wave ripples within the structure, within the hippocampus, and how the secondary, that is the downstream targets, are reading it. And I explained that fed-in readers, such as the lateral septum, care mostly about synchrony, whereas fan-out readers, such as the neocortex, can read out multiple aspects of the sharp wave ripples. And I haven't discussed much about point number 4, but that's also a very critical issue, of course, is how experience can bias what kind of attractors, how many different types, and what are the dominant types, and what are the constellations of spiking activities that are played out after the animal learned the task. And to summarize the last part, it seems that longer ripples are needed for memory, and even artificial prolongation of a ripple can make memory a little bit better. Now, this was the summary of our project, but of course it very effectively dovetails into the presentation of the other members of the Unite team. The replay is being investigated by each of us, and in the next presentation, Attila Losonci will explain how calcium imaging can be utilized to record even larger number of neurons during sharp wave ripples and understand their mysteries. Thank you.
So my name is Attila Losonci, and I'd like to tell you about our recent research on how we use large-scale functional calcium imaging to study neural mechanisms of long-term memory in the hippocampus. So a fundamental function of the mammalian brain is to learn and remember, and our extended human capacity to form and maintain long-term memories largely defines our conscious experience. So long-term spatial and episodic memories are critical for our daily life. For example, we have to remember where we parked our car, not just in a short term, but also in the long-term parking lot at the airport. But how do we form and maintain mental maps of the world around us over a long time scale? A mechanistic understanding of these processes remains a long-standing challenge, and our team is interested in understanding how the neural circuitry in the mammalian, and more specifically in the rodent hippocampus as our model system, supports learning and the formation of long-term memories. So what do we already know about these processes? Let me highlight just a few major findings. The central role of the hippocampus in long-term spatial learning and episodic memory has been long established by human pathophysiological studies, indicating that damage to the hippocampus severely impairs patients such as the famous HM's ability to learn and remember new information about people, places, and events that together constitute our autobiographical memories. Also, a large body of experimental work points to long-term changes in the strength of synaptic connectivity between neurons in the hippocampus as candidate cellular and molecular mechanisms of long-term spatial and episodic memory. Third, we also know about a striking cellular activity correlate of spatial navigation and learning that has been identified across all processing nodes of the hippocampus. And this is the spatially restricted firing of excitatory principal cells at restricted locations as the individual explores the environment. These cells are historically called place cells, and together they tile the surrounding environment and form a cognitive mental map. It is widely thought that specific changes in this cognitive map can encode and anchor spatial, and episodic memories. Finally, as you also heard in the previous talk, hippocampal memory has multiple stages that are associated with distinct network oscillatory patterns that can be captured with local field potential recordings. So memories are initially formed and encoded during exploration, which we can call online activity state of the hippocampus, and this is associated with rhythmic 5 to 10 Hz theta oscillations. To form lasting memories, this initial encoding phase is followed by selective strengthening of memories during a second offline memory consolidation phase that occurs, for example, during immobility or slow wave sleep. Importantly, this consolidation phase is thought to occur over multiple days. And as you also heard in the previous presentation, during this offline state, LFP activity is mostly irregular, but it is occasionally interrupted with large amplitude, short duration population burst events called short wave ripples. And a large body of research has demonstrated that online place cell sequences are reactivated or replayed during these short wave ripples, and this repeated time-compressed reactivation of online sequences are thought to be critically important for long-term memory consolidation. In the previous talk, you heard from Dr. Buzsáky about how ripples can fan out memory-related information from the hippocampus that can be read out by downstream cortical and subcortical targets. In a complementary set of experiments, we asked the opposite questions. Does ripple-related memory reactivation affect the stability of neural representation within the hippocampus? And if so, does it affect all the cells equally? Or it's more important for some, but not the others? A major obstacle to experimentally investigating this problem is that classic electrophysiological recordings cannot reliably track large populations of neurons over multiple days, during which memory consolidation is thought to occur. Luckily, there is a complementary approach, functional calcium imaging, that offers just this ability. So in our lab, we've been using 2-photon calcium imaging to simultaneously track the activity of large number of cells in the mouse hippocampus. This approach allows us to detect changes in intracellular calcium levels that is evoked primarily by changes in neuronal spiking activity, and to measure their spatially localized place cell-like activity at the single cell level, as it is shown on the left. However, we can also reliably track the same neurons over multiple days, which is critical for monitoring memory consolidation. And finally, we can monitor the activity of several hundreds of place cells simultaneously. So Andres Grossmark, a postdoctoral scientist in my lab, who previously used large-scale electrophysiological recordings to study ripples in the Buzsáky lab, sought to take advantage of calcium imaging and combine it with electrophysiology to study neural mechanisms of long-term memory consolidation, and in particular, to answer the question whether ripple-related memory replay affect the stability of hippocampal place cell maps over long time scale during learning. For this, Andres developed a method combining local field potential recordings to detect ripples and simultaneous 2-photon calcium imaging to track the activity of large populations of hippocampal neurons in the mouse. In the learning task, the mouse has to navigate to find a reward location on a virtual reality track during which place cell sequences can be recorded. In addition, Andres recorded and imaged offline activity before and after these learning sessions, during which ripples are particularly abundant. Importantly, these recordings were carried out over multiple days to answer questions about the effect of ripple-related reactivation on longitudinal stability of hippocampal maps. So on each day, individual place cells, such as these 160 simultaneously imaged cells that are shown here, exhibited spatial receptive fields at discrete locations on the virtual environment, nicely tiling the whole navigational track. We then could compare the spatial receptive fields of neurons longitudinally over multiple days. What you see here are neurons that are aligned based on their peak spatial response on day 5. And indeed, what we found that some neurons shown in red stably code for location across days, while other neurons shown in blue either change or lose their place fields, implying the presence of both stable and unstable coding elements in the hippocampal longitudinal memory code. So this was an exciting result. So next we could move on to answer our main scientific question, whether this variability in the longitudinal stability results from differential recruitment of these cells to ripples. And to answer this, we first looked at offline epochs, before and after running, when the animal was immobile and sharp wave ripples occur frequently. So by calcium imaging, as you saw, a powerful method to monitor online place cell activity, we still didn't know if this method also allows us to detect population dynamics during the short duration ripple events. So the movie in the middle shows the population calcium image response of hippocampal pyramidal cells to LFP detected ripples. And hopefully you can see what is actually summarized on the right panel, that LFP ripples are associated with transient, widespread, and large amplitude calcium responses. So these sharp calcium responses confirm that our imaging method has the required temporal resolution to study the fine time scale of ripple-related neuronal reactivation. What was even more exciting to us that using calcium imaging, we were able to observe that within these individual population events, place cells reactivated in the same sequence as observed during the previous running bout. Here are two events showing forward replay of place cell sequences during ripples on the left and reverse replay on the right, similar that you have seen in the previous presentation using electrophysiological recordings. Here you can see another 6 example sequences from pre, run, and post parts of the learning trial. By quantifying the amount of sequenceness associated with ripples, we found that place cell sequences were enhanced during and after goal-directed learning behaviors, as shown on the upper right, consistent with the learning dependency of ripple-related memory replay. So one take-home message is that cellular resolution calcium imaging is a powerful tool to study memory replay. Now with these methods in hand, we can directly examine the putative role of ripples in memory consolidation. And we did this by testing whether we could use individual place cells learning-related changes during the early phases of the learning to predict their future place coding stability. As the bar plot on the left summarizes, we indeed found that cells that were preferentially recruited to ripples, or in other words, cells that we call positive ripple gain cells, were associated with increased long-term stability. This means that we were able to directly observe for the first time the role of ripples in multi-day memory consolidation. Interestingly, however, we also found that the stabilizing effect of ripple recruitment was the largest for place cell coding far from the reward. The plot in the middle and the right show the stability of cross-day spatial coding as a function of position in the virtual environment for cells that had negative or positive ripple gain. What we found was that the negative ripple gain group in the middle panel showed a specific decrease in cross-day stability farther from the reward location in the middle of the track. In contrast, cells that were recruited to ripples were stable both near and far from the reward, as is shown on the right panel. Of course, what you may have guessed already, during this learning task, the animal likes to spend more time at the reward location, just like we ideally spend more time in our favorite restaurant when we go out and not sitting in a traffic. So therefore, our result is consistent with a model in which hippocampal memory consolidation actively compensates for reward-skewed behavioral sampling of the environment and promotes the stabilization of spatially accurate and more useful long-term spatial maps. Now you may also wonder if these results actually make sense. Don't we actually want to preferentially remember important events and locations, such as the reward in this task? Maybe the next example can help to contextualize our findings. When we move to a new city, we initially form a map that contains a list of most important locations, for example, pizza joints, a subway station, or the Central Park in New York City, but with very little details between these locations. With experience, we form a finer grained, high-resolution representation of the city that we can also use to chart new trajectories and shortcuts between them. And for this, we also need to represent the actual path and trajectory between them. So the major take-home message of this talk is that our results indicate that hippocampal ripple-related memory replay actually helps to form and consolidate long-term memories that include both salient locations and the path between them in a mental map. Finally, I also hope to convince you that functional calcium imaging is a powerful approach to answer questions about memory replay and consolidation. And in the next presentation, Dr. Mark Schnitzer, another member of our team, will tell you about how we can also develop and use even larger scale imaging methods across the whole cortical mantle and to directly image voltage signals in the brain to study memory. Thank you.
I'm Mark Schnitzer of the Departments of Biology and Applied Physics at Stanford University, as well as the Howard Hughes Medical Institute. And today I'm going to tell you about how my lab has been developing and applying optical approaches for studying how the brain stores and recalls long-term memories, together with my collaborators as part of an NIH Brain Initiative U19 grant project. Now, for many studies of long-term memory, we would like to be able to combine different methods that together will allow us to track the activity of large neural populations over extended time periods pertinent to long-term memory. First, we'd like to be able to image neurons, regardless of whether they might lie in deep or in superficial brain areas. We would like to perform time-lapse imaging experiments of these neurons over days and weeks over the course of a long-term memory experiment. We want to be able to watch the dynamics of these cells in freely behaving animals, performing natural behaviors. Together, these approaches often produce very large data sets on the order of tens of terabytes or more. And it's important that we have computational analytic approaches that are up to the job of analyzing these large data sets. And as I will seek to illustrate today, the combination of all these different methods has afforded us new possibilities for studies in which we can track the dynamics of large neural populations over extended time periods in the context of a memory experiment. And I'm now going to introduce you to these elements individually and show you how we can apply them. So a number of years ago, we developed implantable optical microendoscopes or optical microneedles that can provide us micron scale resolution of neurons residing deep within the brain of live animals. And here you can see two different examples of these optical microendoscopes. The one on the left is 1,000 microns in diameter. This one is 350 microns in diameter. At the tip of each, you can see a little micro-optical objective lens that gives us the micron scale resolution we need to see individual cells. And it's followed in each case by a much longer but weaker relay lens, whose main purpose is to give us the reach we need to penetrate into deep brain tissue. Now, these endoscopes can be permanently implanted into the live adult mouse brain. And to illustrate here, I'm showing you some time-lapse image data sets taken from the CA1 area of the hippocampus in a live adult mouse that is expressing a fluorescent marker in CA1 hippocampal pyramidal cells. And you can see that using these implanted microendoscopes, we can come back again and again over periods of 2 months or more to these individual neurons, as shown here on the left. And as shown in this middle column, you can see that we have the precision of return to come back again and again to individual neuronal dendrites. Now, for our U19 project, we are applying this approach in collaboration with Ivan Solstis's lab to be able to image neurons repeatedly in the supramammillary nucleus. This approach even gives us the precision to track individual synapses, as illustrated here, again, using fluorescently labeled hippocampal pyramidal cells. You can see from these images in this time-lapse data set that we can come back to individual dendritic spines, as indicated with these arrowheads. And these are postsynaptic morphological structures on the vast majority of excitatory neurons that these CA1 pyramidal cells receive. And you can see over this 3-week or so period that some of the Dendritic spines and hence synapses persist over the course of the experiment. New spines arise midway through, other spines disappear, and yet another population is recurring throughout the experiment. And so you can see we have very good precision of return to individual neurons and individual neural subcellular elements. Another important tool for us in our studies of memory is a miniature optical microscope that has a mass of about 2 grams. It's based on semiconductor optoelectronics and is small and light enough that it can be mounted on the head of an adult mouse as it naturally behaves. My colleagues and I at Stanford originally developed this technology, and we have since commercialized it through a company known as Inscopics. There are over 400 labs using this approach worldwide and over 1,000 microscopes in the field. And for financial disclosure, my colleagues and I continue to have a financial interest in Inscopics. This slide shows you a little bit more about how a microscope works and the parts that are inside. The microscope is used in conjunction with a microendoscope that is implanted into the brain area of interest. Here on the right, you can see a magnified view of the miniature microscope. There's a micro-optical objective. There's a miniaturized fluorescence dichroic filter set, which resides here. On the side, there's a printed circuit board which carries LED illumination. And on the top, there's another printed circuit board that carries a cell phone CMOS camera for the acquisition of images. This technology was originally the product of 3 different PhD dissertations at Stanford, done in collaboration with my Stanford colleague, Abbas Al-Gamal. And it was the 5 of us that originally started Inscopics, as I mentioned earlier. This slide gives you an example of the miniature microscope at work. On the left, we see an adult mouse wearing the miniature microscope. It has a microendoscope implanted in the CA1 region of hippocampus. And on the right, you can see neural calcium activity in the cell bodies of CA1 pyramidal cells expressing a genetically encoded calcium fluorescent indicator. And so those fluorescing spots are cell bodies that are firing bursts of action potentials. We can track the neural activity concurrently with the animal behavior. We're seeing the activity patterns of about 700 neurons at once. This slide shows you that we have the ability to identify the locations of hippocampal neurons as well as their corresponding time traces of activity, not shown here. This is a very favorable mouse. In fact, it's our best one ever, in which we were able to extract over 1,200 CA1 pyramidal cells from one mouse in a single imaging session. And the cell bodies are shown here in red, overlaid a green image showing the fluorescence background from the brain tissue. When we record the activity patterns of hippocampal neurons in this way, we see their typical place coding properties, as has been well documented in the scientific literature. This is illustrated here using 2 different enclosures or arenas, a square box and a circular box seen from a bird's eye view. And when we look at the locations of the mouse in the box and how it relates to the activity patterns of individual cells, we find that the individual neurons preferentially fire when the mouse is in one location of the box. So let's take a look here at cell 348. When we place the mouse in the square box, this neuron preferentially fires when the animal is in the south. But if we pick up the mouse and move it to this circular box, you can see that cell 348 preferentially is active when the animal is in the northeast. And not every cell is very active in all the boxes, and they can often have different locations that are preferred, as you can see here for cell 348. Cell 159 is active generally in only one of these boxes. It likes to fire in the northwest of the square box, but not so much in the circular box, and vice versa for cell 198. Preferentially fires here in the southwest of the circular box and not so much in the square box. And these kind of remapping qualities are well established in the literature, but now we can see them optically in freely behaving animals. We can have many microscopes working at once, as illustrated here in this montage of brain videos acquired from 5 different mice. The microendoscopes in this case have been implanted in the dorsal striatum. And this slide illustrates that even when the course of a single afternoon of experimentation, we can acquire very large data sets in the range of terabytes, tens of terabytes or more. So it's very important that we have a computational pipeline that is up to the job of analyzing all this data. And over the last several years, my colleagues and I have developed a substantial body of code that allows us to do this. This pipeline is schematized here, and I'm not going to go through all the details, but we have different modules, one which is dedicated to inputting the video data, another dedicated to pre-processing the video data. This third module is perhaps the most important. This is where we extract the identities of the individual neurons and their corresponding time traces of activity. And here in this fourth module, we can register the cells and their identities across the multiple different days of a time-lapse imaging experiment, allowing us to track faithfully individual neurons over extended time periods. I'm now going to illustrate how we can use our computational approaches to extract new information about memory. So here's a case in which we trained a mouse to run back and forth in a linear track, an environment that was very familiar to the animal. The black traces show the animal's actual passage back and forth through space. By extracting the activities of individual neurons and using machine learning methods, we can deduce from the calcium video data sets alone where the animal was in space. And so here you can see from the red traces the position of the mouse as we would predict based on the activity data from the hippocampus. Now we can also test how well this representation of space in the hippocampus may be accurate. How long does the mouse maintain this neural representation of space, akin in some regards to a spatial memory? We can perform this test by constructing the decoder on one day and testing its performance when the mouse runs the linear track on a different day. So here you can see data in this top row from a 5-day interval between the construction of the decoder and its application to predict the mouse's location 5 days subsequent. Here in this middle row, you can see the same kind of test with a 15-day interval between the construction of the decoder and its testing. And the decoder has indeed lost some of its accuracy, but is still very accurate. Down here in the bottom row, we can see results from a 30-day lapse between the construction and the testing of the decoder. The decoder indeed has, again, lost some fidelity over these shorter intervals, but nevertheless is making predictions about the mouse's location that are highly statistically significant. In more recent experiments, we can now begin to track how the animal and the hippocampal neural dynamics will learn the representation of a new spatial environment in the context of a long-term spatial navigation and memory task. Here you can see a movie on the left in which a mouse is navigating a 8-arm radial maze. One of the arms has been baited with a reward for the animal. On each trial, we start the animal in one of the other 7 arms. The animal has to look at some distal spatial cues, which are not visible in this video. They're on the wallpaper of the arena. And based on these cues, the animal can navigate to the goal arm and receive its reward. As the animal learns to do this over multiple days, we can track the patterns of hippocampal neural activity and extract the hippocampal representation of space and begin to examine the manner and the time course with which the hippocampus will learn this radial maze and relate it to the animal's progressively improved performance. So I hope to have illustrated for you now that by combining a variety of different methods, we have promising new capabilities for tracking neural ensemble dynamics and the information that they encode over the course of long-term experiments regarding memory, storage, and recall. In the last portion of the talk, I'm now going to show you how we are creating new optical technologies that my colleagues and I will be using for studies of memory as part of our U19 project. To summarize, we are developing optical techniques that will allow us to image more than one brain area at a time and to see how different brain areas can cooperate together in the service of long-term memory. We are also working with techniques that allow us to perform causal manipulations, optogenetic manipulations at the site of imaging in the brain. I'll show you how we are developing and using techniques for imaging neural activity across the dorsal mantle of the cortical surface. And finally, I will discuss some techniques that allow us to track neural activity by using the transmembrane potential of neurons rather than the concentration of intracellular calcium as a proxy for neural activity. With these newer methods, we can track neural voltage dynamics directly. So let's look at these individual new emerging technological approaches. So first, if we want to be able to image the dynamics of more than one brain area at a time, one simple approach is to implant more than one miniature optical microscope. And here you can see a mouse that is navigating a 4-arm plus maze as it wears 2 different microscopes, one that is allowing us to track the somatic calcium dynamics of neurons in the prefrontal cortex, while the other microscope is allowing us to track somatic calcium dynamics of hippocampal pyramidal cells. And so in this way, we can visualize and analyze how these 2 different brain areas may be coordinating their activity patterns as the animal performs this spatial navigation task. The most recent versions of the miniature microscope incorporate 2 different light sources, one that permits optical imaging of calcium activity and another that permits optogenetic manipulations of neurons under the same field of view. To access neural dynamics across the dorsal surface of the mouse neocortex, we have developed a mouse preparation that we call the crystal skull. In this approach, we permanently implant, in place of the natural cranium, a curved glass window that provides long-term optical access to the cortical surface. Here you can see some examples of the use of the crystal skull. This is a transgenic mouse made by Anqi Zhang and her colleagues at the Allen Institute. This animal expresses a genetically encoded calcium indicator in layer 2-3 neocortical pyramidal cells. This mouse is running, and using a fluorescence macroscope, we can track somatic calcium activity across the cortical surface. If we zoom in, you can see more clearly the activity patterns of individual neurons. Each one of those fluorescent spots represents an individual neuron firing probably a burst of action potentials, and we can zoom in even further. This panel here on the right shows you a magnified view of this inset here, and we can very clearly see the activity patterns of the individual cells as well as the hemodynamics through the different blood vessels here. So through this crystal skull, we can perform a variety of different optical imaging techniques. Some of the images that result from both 1-photon imaging, 2-photon fluorescence imaging here are shown in this set of images. For example, here is a montage of pyramidal cells in a coronal view. Comparison to the Allen brain map shows that we can sample cells in about 36 to 40 brain areas across an individual animal that is wearing a crystal skull. And we are now using this technique in collaboration with Yuri Bazaki's lab to be able to track patterns of neural activity associated with sharp wave ripples. As you can imagine, as we gather information about the dynamics of neurons across so many different brain regions at a time, our computational pipeline is absolutely critical. And by refining it to work quickly and on high-performance computing clusters, we can now analyze data more quickly than we can even acquire it. So this is an example in which we have distributed the code across 56 different computer cores to analyze data sets covering multiple brain areas at a time. Finally, I'm going to tell you about work developing and applying fluorescent optical voltage indicators that allow us to track the transmembrane potential of neurons directly rather than the intracellular concentrations of calcium and the rises in calcium that are associated with spiking. Now, using voltage indicators, we can track action potential dynamics and membrane potential dynamics directly. Our efforts in this regard have focused upon a particular class of fluorescent voltage indicators known as Fret-Opson voltage indicators that allow us to detect individual action potentials with high signal-to-noise ratio using only milliwatts of light. And here, to illustrate, you can see an electrophysiologically recorded trace from one neuron. At the same time, here in the blue, the optical trace recorded from the same neuron. And you can see that the two traces match very well. We catch each and every spike with the optical voltage indicator. We have verified the fidelity of this approach in the brains of live mice using simultaneous electrical and optical recordings. In this case, we're looking at an optical recording from a particular voltage indicator known as H2N M neon green. We performed concurrent loose patch electrical recordings and optical voltage recordings and showed that we could catch each and every spike in the optical traces. Moreover, using the electrical records as the ground truth, we found that we could time the spikes from the optical records to within plus or minus 200 microseconds spike timing precision. Together with collaborators at Yale, we have been exploring new versions of the fluorescent optical voltage indicators with different fluorescent colors. We have a red version of the indicators. We have a green version. The red version is known as Varnum and was published in 2018. And we can now therefore look at the voltage dynamics of two different cell populations at once. And one way we're exploring this is with a fiber optic measurement technique that we call TEMPO, which stands for transmembrane electrical measurements performed optically. And I won't go into all the details of how this approach works, but it is designed to allow us to look at optical voltage rhythms, a phenomenon which is critical to our U19 project on sharp wave ripples. This measurement technique actually attains the physical limit on sensitivity that is set by photon shot noise, and improvements thereby will come, for example, with improved indicators over time, but the instrumentation is already reaching the physical limits on sensitivity. TEMPO has several advantages over conventional electrical field recordings for sensing the patterns of voltage oscillations in the live brain. First of all, TEMPO allows us to target with the genetically encoded fluorescent voltage indicator specific cell types of interest. This is not possible with electrical field recordings. Moreover, the interpretations of our recordings are substantially improved in the sense that we know we are monitoring transmembrane voltage directly. We do not have to worry about interpretive difficulties from volume conduction effects, as is often an issue with electrophysiological field recordings. The specific neuron types that we target with our voltage indicator can be sparse, such as different subclasses of interneurons. We often have superior sensitivity to electrofield recordings in non-laminar areas of the brain. And overall, with TEMPO, there are new opportunities to dissect the different contributions of selected cell types to the voltage rhythms that we might see with electrical field recordings. So here's an example in which we use the red fluorescent Varnum indicator targeted to hippocampal pyramidal cells. The traces on the top here are from a running mouse, and you can see that the theta rhythm in the 6 to 10 hertz range that we've recorded with the optical approach corresponds very well to what you see with a local field potential recording near the tip of the optical fiber that's been implanted into the hippocampus. And we see the same level of concordance here in the delta band when the mouse is not moving. The optical approach works very well at higher frequency bands. For example, these are recordings of a sensory evoked neocortical gamma oscillation. And with our colleagues, we are working on extending this approach to the sharp wave ripple band. Now, one of the important aspects of TEMPO is that it offers the possibility of dissecting different cell type contributions to the brain's oscillatory phenomena. And here you see this illustrated with a delta rhythm. In one case, we've targeted the indicator to pyramidal cells, shown here in the orange trace. In another case, we've targeted the indicator to parvalbumin positive interneurons, shown in the green trace. And we are referencing these oscillations seen optically through the local field potential seen here in the blue traces. The frequencies of oscillation in the different traces are the same between this example and this example, but they're different phase shifts, suggesting that the pyramidal cells and the interneurons can play different roles in constructing the field oscillation. And so we hope to do more of this going forward, particularly in the context of sharp wave ripples. We're going to be applying this approach again in collaboration with Yuri Bazaki's lab as part of that effort within our U19 project. So in closing, I'd like to thank all of the lab members and collaborators that contributed to different aspects of this work, and to the different funding agencies that made this work possible, in particular, the NIH Brain Initiative that is underwriting our collaborative U19 grant project. Thank you for your attention.
Hello. Let's now talk about some of the inhibitory interneuronal microcircuits that may underlie the generation and mechanisms of sharp wave ripples and multi-scale modeling that we use to understand them. So far, the emphasis has been on excitatory circuits within the hippocampus to a large extent, such as the granule cells of the dentate gyrus, CA3, of CA3 pyramidal cells and CA1 pyramidal cells. These major cell groups, of course, are connected via excitatory synapses. However, in a brain region such as the CA1, but this is true for any cortical region, in addition to the excitatory glutamatergic cells, there are also a lot of other cell types that are very diverse, and they come in different shapes and sizes, and we refer to them as GABAergic interneurons. As you can see on this slide here, we distinguish within the CA1 something approximately about 20 to 30 interneurons. You see 15 of them listed here. These are the most important types, but there are others as well. And these interneurons are classified based on their input-output characteristics, where the soma is located, where the dendrites are, and which part of the pyramidal cells the axons innervate. The pyramidal cells themselves are not indicated in this figure, but they would be located in the stratum pyramidale layer that's indicated as SP in this drawing. So you see, for example, that there are about 9,000 parvalbumin positive interneurons indicated in red at the bottom in the CA1 of a rodent. And they consist of various subtypes: exoaxonic or chandelier cells that innervate the initial segment of pyramidal cells, basket cells that innervate the perisomatic region, and bistratified cells that innervate the proximal dendrites. There are also other types, for example, neuropeptide Y positive cells, or somatostatin positive cells, or cholecystokinin positive cells. And these are all labeled here and indicated as color, and the numbers are also indicated. Now, in the CA1, we have about 300,000 or more CA1 pyramidal cells. So these GABAergic interneurons are few in numbers relatively to the much more abundant pyramidal cells, but they play an incredibly important role. As you see on the right, and I'm just going to use here the example of the parvalbumin and CCK basket cells, these cells, these interneurons, are not just different in terms of their dendrites and axons, their morphology, but in fact they are different in all kinds of other ways, molecularly, and their connectivity and their properties as well. So let's just discuss this for a minute. For example, the parvalbumin cells they receive a lot of excitatory inputs, as you see, feed-forward glutamatergic inputs as well as feedback glutamatergic inputs. They fire very fast action potentials in a non-accommodating manner. Their time constant, membrane time constant, is fast, so they are really made to process signals very fast at high fidelity. And even when you look at their axonal terminals, what you see is that when they release GABA, they release the neurotransmitter GABA, they cause postsynaptic inhibitory events in the pyramidal cells with high fidelity. In other words, whenever there is a presynaptic action potential, we see a temporally tightly coupled postsynaptic response. Contrast this with the CCK basket cells, and those are also basket cells. They innervate the perisomatic regions. But these CCK or cholecystokinin positive cells could not be more different than the PV cells, from the PV cells. Indeed, they receive relatively few excitatory inputs, glutamatergic excitatory inputs, and they fire regular spikes, slower spikes that do accommodate. They have slower membrane time constants, and then when they release GABA, they generally release GABA in what we call an asynchronous manner, as is indicated on the bottom little square there. So each of these interneuronal subtypes really are little machines that are connected in the circuit differently, and they express a variety of sodium and potassium channels and calcium channels that are very specific to the interneuronal subtypes, as well as other neuromodulatory receptors distributed across these different cell types in a very cell type specific manner. So how do these interneuronal subtypes behave during ripples? That's really the question here. So I'm going to show you some data from my lab that we obtained using what we call juxtacellular recording techniques. These are techniques where we, in vivo, we approach a single cell with an electrode, and we record a single cell, and then we feel it, and that cell's activity is recorded, and then we post hoc verify that indeed that cell was, for example, a parvalbumin basket cell that you see up there with the red dendrites and the blue exons. And then in some cases, we also show that they are parvalbumin immunoreactive, and we use electron microscopy to verify that indeed these parvalbumin basket cells form synapses on somata. At the bottom, you see the so-called OLM cells, which is named after the fact that the cell body is in the oriens layer, and it projects to the lacunosum molecular layer, the distal dendritic layer of the CA1, where the temporal amino fibers come in from the entorhinal cortex. So these are the OLM cells. So these are two different interneurons. One of them is a perisomatically projecting GABAergic inhibitory neuron. The other one is a distal dendritically projecting interneuron. And here is how they fire during ripples that are simultaneously recorded with local field potential or LFP electrode. Just by eye, you can tell that the parvalbumin basket cells fire on most ripples, or if not all of the ripples, at high frequencies, whereas the OLM cells fire on, let's say, 50% of the ripples, and when they do fire, they don't fire as much. And indeed, this is quantified here. Now we can actually dig into the ripples even more, and we can look at how these two cell types fire during individual ripple wavelets, if you want. And when we look at that, we can see that, for example, the parvalbumin basket cell, as you can see in panel E, indicated by the red arrow, tend to emit action potentials preferentially during the rising phase of the individual ripple wavelets, whereas the OLM cells, the dendritically projecting OLM cells, tend to fire a little bit later. So this is shown here in a more easily digestible cartoon format. You see that the red cells, the parvalbumin basket cells, project to the perisomatic region, and they tend to fire during the initial rising phase of these ripple oscillations, whereas the green cells, the OLM cells, tend to fire later. So this is a temporal ordering of perisomatic and dendritic inhibition coming from these two cell types. And you can think of ways why this would happen. The CA3 inputs that's indicated there is exciting, for example, the PV basket cells and the pyramidal cells, but not the OLM cells whose dendrites are down in the oriens. So therefore, the orient cells will receive excitation only once the pyramidal cells are firing. So that may explain this delay. Interestingly, we notice that such a temporal ordering is present at other frequencies as well, as you can see here. So this is an example of frequency invariant temporal ordering of perisomatic and dendritic inhibition, because, for example, during the theta oscillations that occur when the animal is exploring or moving, these theta oscillations can also be picked up by these LFP electrodes, and they occur at slower frequencies, about 4 to 10 hertz. And as you can see still, the parvalbumin basket cells fire first, followed by the OLM cells. Another example that I'll show here is that even when you dig into the parvalbumin family of cells, I mentioned that the parvalbumin family cells, they comprise of the exoaxonic cells that innervate the initial segment. You see them in panel A in blue. Then there are the basket cells that we just discussed in red. And then there are these bistratified cells that are shown here in green. And by eye, if you look at these recordings, you can see that the exoaxonic cells tend to fire the earliest during the falling phase of the oscillations, followed by the parvalbumin basket cells, followed by the bistratified cells. And you can also see that the exoaxonic cells, not only that they fire first, but they fire very tightly temporally logged. They are strongly modulated by the theta rhythms, whereas the other cells are modulated less so. So what these results show, for example, is that during every single theta wave, there is a sweep of inhibition coming from these parvalbumin family cells, from the initial segment to the soma to the dendrites at each and every theta wave. So we of course collected data from many other interneural subtypes that I don't have time to show here. What we can do with all of these data is that we can actually assemble this into a multi-scale model, a computational model. Let me just talk a little bit about this. Of course, I should say that much of the data is not just coming from my own lab or my collaborators' lab, but through the entire neuroscientific community, since the hippocampal networks, and in particular the dentate and the CA1 networks, are very well studied by other labs all over the world. So these models, we have, what we have in these models is we explicitly represent these various interneural subtypes. As you can see in panel D, we have all of these interneural subtypes at the right proportion. Each cell number is determined, and the connectivity database was published separately as indicated there. And then we connect these cells through realistic synapses with realistic synaptic numbers. Each cell expresses ion channels based on experimental data, as is shown on the right, upper right, with ion channel distributions along the dendrites. And the ion channels, for example, sodium and potassium and calcium channels, generate cell type specific firing properties that you see here on the right, color coded. For example, in the top, you see in greenish color, you see the fast spiking basket cells, and in yellow, you see the CCK basket cells that we discussed. So then we have these cells distributed in 3D in realistic numbers and coupled to each other and to the pyramidal cells in realistic ways. And so the question is, what happens if this network is we just see what this network can do? First, we can look at this network and just decouple it from the rest of the network, so we don't drive it with CA3 or entorhinal or medial septal rhythmic inputs. And the question is, what is it that this network, through the interaction of excitator and inhibitor cells, what is it that it can generate? What are the emergent properties of this network? What you can see is that when you drive this CA1 model, this large scale, actually full scale, multi-scale model, multi-scale meaning from synapses and molecules, ion channels, all the way to the whole network. What we see is that theta oscillations and phase-logged gamma oscillations appear, as you can see in A and B. Gamma oscillations occur around 25 to 100 hertz. And we know that they normally occur at certain phases of the theta rhythm, and that is actually captured by these models. In addition, when we drive the CA1 model with a particular distributed Gaussian distributed excitatory wave representing the sharp wave depolarization of the CA1 dendrites, we can actually see ripple oscillations appear as well. And then in the model, of course, we can look at how the various interneurons fired at any of these rhythms, and we can study whether they match our experimental observations. And indeed, we can, for example, see, as is indicated on the right, that when you compare the model prediction with the actual experimental data, and here each arrow is the difference between the firing of the model interneuronal subtype, again color-coded. The greenish one is, for example, the parvomine basket cell again, and the yellow one is the CCK basket cell, etc. So we can compare them, and the size of the arrow represents the deviation of the model prediction from the actual data gathered from animals. And you can see that by and large, there's a very good agreement between the model and the data regarding the phase-specific firing of the various interneurons during these slow theta oscillations. And then you can also do various interesting optogenetic, in silico optogenetic manipulations, such as shown on the left under B. So for example, in control animal, you see a certain amount of theta power. And then when we inhibit in the model the parvomine basket cells in a virtual optogenetic experiment, you can see that the theta power collapses again in agreement with experimental observations. And I should say that these modeling results are not just interesting when there is agreement between model and data, but the outcome where the model doesn't agree with the data is equally interesting, because then we can dig into that and try to understand why there is a disagreement, and we can then carry on with new experiments. So this was all about oscillations, but we can also model, and we are starting to do this more and more, spatial feature selectivity. And let me explain to you what I mean by this. So with experiments, we can focus on certain cell types. And I'm going to bring in here the so-called mossy cells that have been of great interest to me and to many others, which are actually excitatory cells. In a way, you could call them interneurons, since their axons don't leave the hippocampus. These are excitatory cells located within the dentate gyrus, and they innervate granule cell and interneural dendrites. And interesting feature is that in many neurological disorders, such as epilepsy, ischemia, and others, these mossy cells are selectively vulnerable. So that has triggered a lot of interest in these cells. But what do they do in the normal brain? In 2017, 3 papers appeared, 2 from our collaborators, our team members, from Gyuri Buzsáki's lab and Attila's lab, in Neuron, all showing basically that the dentate gyrus mossy cells in behaving mice are very sensitive in their so-called place field activities to small changes in the environment, as that's shown in that diagram from Gyuri's work. And you can see that even compared to the granule cells, where these place fields of granule cells don't remap much in this particular experiment, whereas the mossy cell place fields are preferentially remapping or changing when the animal is moved between environments. And these results from these 3 labs, from the Buzsáki, Losonci, and Jim Knierim's labs, all pointed to a potential role of mossy cells in spatial coding. And indeed, then we tested this idea: do they actually play a role in spatial memory encoding? With Enbu in my lab, and this is shown here. So if you take an animal where we can manipulate mossy cells, inhibit mossy cell activity when we want to using optogenetics, and you put a normal animal, an animal here without the light on, without inhibiting mossy cells, what you see is that in this particular paradigm, during training, which lasts about 10 minutes, this is called the object location memory test, animals sniff those 2 objects roughly equally. They are curious animals. And then you can take the animals out for a day and then put that back during the testing period. This is under panel C here, for a few minutes, 5 minutes in this case, with 1 object moved. And normal animals, and this is what you see here on the right in gray bars, either in the dorsal or ventral so normal animals, as shown with gray, will sniff the moved object much more. They are curious. It's a little bit like you go home and you see that your favorite chair is moved in the room, you are going to look at that more. Similarly with mice. So normal animals are kind of remembering where that object was. This is why it's a test of spatial memory. However, the interesting thing comes that when we inhibit MOSI cells during the training, during the learning period, what we see is that the mice don't differentiate between the moved and the unmoved object. This is shown on the right with those orange bars that are basically at chance level. They are not differentiating between the moved and the unmoved objects, as if they don't remember the spatial position of the original spatial position of the objects. And it doesn't matter whether we inhibit MOSI cells in the dorsal or ventral hippocampus during training. However, notice that under D, when we inhibited MOSI cells during testing, then there was no effect. The mice performed with the MOSI cells decoupled from the network just as well as control mice. So this is an example of how we can dig into specific cell types in terms of their role in spatial memory encoding, which is, of course, very important for the ripple-related memory replay. So we can actually dig into this even more using modeling techniques that I talked about. So in this case, we are building a large-scale, multi-scale model of the dentate gyrus with all those interneurons and the granule cells, the pyramidal cells, so the granule cells, the principal cells of the dentate gyrus, as well as MOSI cells that you see in red in the hilus of the dentate gyrus, with the right connectivity again. And as you see in the bottom left, each granule cell has its own unique dendrite, and these granule cells are distributed in 3D, as you see at in the middle in that 3D image. And these cells are then connected in a cell type specific and distance dependent manner, as is indicated on the right. You are now looking at the flattened out dentate gyrus network. And we can model the topography of the spatial inputs to the hippocampus from the entorhinal grid cells. That we know, as shown on the bottom left, that the grid cell spacing change as a function of the dorsoventral position in the entorhinal cortex. These fibers go into the dentate gyrus, and we represent them explicitly. And then we run the simulations using a virtual mouse running through the field and excite various entorhinal grid cells in a 2D spatial rate map, as shown there. And then the question is, do we get an emergent place cell activity in granule cells and MOSI cells? And the answer is we do, then that's shown on the right. And then we can study what proportion of granule cells, for example, have no place fields, what proportion have 1 or 2 place fields, and what is the information content of these place fields. And we can compare them to any other cell type, in this case, the MOSI cells, but we can also do that with in the CA1 and ask similar questions elsewhere as well. All of these modeling in these multi-scale, very large model networks is a difficult enterprise, and we use supercomputers for this, funded by NSF funds, such as funding mechanisms, such as the XSEED program. That is an excellent program, and we can also develop. We do develop new modeling tools, such as what is written here as the multi-objective network optimization framework by our team. Mostly led by Aaron Milstein and Ivan Rykov. And the idea here is to really solve this big problem of how you optimize such a large network with hundreds of parameters, ranging from channel densities of various ion channels to synaptic strengths and synaptic kinetics, etc. And as you can see, the idea here is to run each model through a battery of simulated experiments in parallel and evaluate the multiple potentially conflicting objectives and compare to experimental targets, such as input-output slope, spike rate adaptation, attenuation from dendrite to soma, synaptic integration and others, and at each iteration, choose new models based on past model performance. So we are trying to work on these new methods to optimize these very large frameworks, networks, to actually arrive at a really good representation of these hippocampal networks and get at the sharp wave memory generation and replay and the mechanisms, cellular synaptic mechanisms that underlie all of these events. Now, it's an important problem that our labs should be able to freely exchange data. But exchanging data is only possible if there is a shared specification for neuroscience data. And there is a large effort, that's an international effort, going through many labs, and we are part of that, led by Ben Dichter, our data scientist. And this is called the Neurodata Without Borders effort. And you see that the idea here is to use, for example, electrophysiology data, like multi-unit arrays from Judy Bujaki's lab, or optical physiology from Attila's and Mark's labs, and simulation data from my lab and other labs, and to really to be able to freely exchange these data using a common formats. So there are NWB data storage rules, as you see on the right, input-output tools and rule enforcement, and various analysis and visualization tools and rules that really help the seamless integration of this team effort through between labs, between the team labs, as well as with outside labs. And this really helps not just data sharing, but also tool sharing, data archiving, and reproducibility as well. So I hope you have enjoyed this webinar from LabRoots, and I hope that you were convinced, you are now convinced of the extreme value of team science that made all of this advance possible in such a short period of time. So a big shout out and thanks to NIH for the BRAIN initiative. Thank you.